Action potentials occur only in axons, and the rate of the action potential propagation depends on two factors. The axon diameter, because the larger the diameter, the less resistance to the current flow, and also the degree of myelination. So if you look at an area that's not at the axon, so another process, one of the other processes like the dendrites, there's going to be um, these are areas without voltage gated channels. So the voltage decays because the current leaks across the membrane. So no action potentials occur at the dendrites. If you look at a non-myelinated axon, so we are looking at the axon here, but they're non-myelinated, there's going to be voltage gated channels like sodium and potassium channels that regenerate an action potential at each point where the channels are located. So the action potential happens, but it's slow. The conduction is slow because it takes time for those ions and for the channels to to open and allow for those ions to come in or out. If you compare that to a myelinated axon, so these are the axons with the myelin sheaths, and if there's myelin sheaths, there's going to be the myelin sheath gaps or the nodes of Rambier. This is going to be the area where the voltage gated channels are located, and that's going to be the area where the action potentials happen. So that's going to allow for the signal to jump from gap to gap, which will allow it to go a lot faster than if it's traveling this way, like the unmyelinated axon. So it'll be about 30 times faster by being able to jump from gap to gap. The synapse is where two neurons meet. So it's where one neuron is able to send information to another neuron. And so there's presynaptic neurons and postsynaptic neurons. So these are just relative positions because presynaptic neuron, one could be a presynaptic neuron in one situation, but it could be the postsynaptic neuron of another. It depends on how the signal is traveling. Okay? But the presynaptic neuron, that's conducting impulses towards the synapse, so towards the meeting of the two neurons. It's sending the information. The postsynaptic neuron is receiving that information. And in the peripheral nervous system, the postsynaptic neuron could either be a muscle cell or a glial cell, or it could be a neuron. So here you could see the meeting of these two neurons. So in this case, this would be the presynaptic neuron that's connecting to another neuron because it's connecting to the cell body of a neuron or the soma. And in this situation, this one would be the presynaptic neuron, and this one would be the postsynaptic neuron. Because in this case, the signal is going from this neuron, and this one is sending the signal to this neuron. So this is the one that's receiving it. So the postsynaptic neurons are going to be the ones that receive the signal. If you're looking at these two, then this one's the postsynaptic neuron because it's receiving the signal that's coming from this one. This one is the presynaptic neuron because it's sending the signal. If you're looking at these two, this one's the presynaptic neuron because this was the one that's sending the signal, and this one would be the postsynaptic neuron because it's the one that's receiving the signal. So the most common type of synapse are chemical synapses. These are specialized for the release and the reception of chemical neurotransmitters. And they're typically composed of two parts. There's the axon terminal of a presynaptic neuron, which contains the synaptic vesicles filled with the neurotransmitter. And then there's the receptor region on the postsynaptic neuron's membrane, which receives the neurotransmitter. So these two parts are going to be separated by a fluid-filled synaptic cleft. So there's a space, a fluid-filled space between the two neurons. And the electrical impulse is going to be changed to a chemical signal across the synapse. But then once it reaches the other neuron or the postsynaptic neuron, it'll turn back into an electrical signal. So we have an action potential coming from, let's say, this neuron here, the presynaptic neuron, because this is the one that's sending the signal. So that signal is traveling along the axon of this neuron. And then the action potential is going to arrive at the axon terminal. There, the action potential is going to open the voltage-gated calcium channels. And then calcium is going to enter the neuron. Okay, because calcium is going to be higher outside of the neuron than it is inside, so it'll travel along its concentration gradient, so it'll move inside. When calcium moves in, it's going to cause the synaptic vesicles to release the neurotransmitter. So all of these are synaptic vesicles, and they contain the neurotransmitters. So when calcium enters, it'll cause those synaptic vesicles to release the neurotransmitters. And the neurotransmitter is going to diffuse across the synaptic cleft. So this is the synaptic cleft here, okay, between the two neurons, the presynaptic neuron and the postsynaptic neuron. So the neurotransmitters are going to diffuse out of the presynaptic neuron into the synaptic, across the synaptic cleft, and they're going to bind to specific receptors on the postsynaptic neuron, which are here. Then once those neurotransmitters are bound to those receptors on the postsynaptic neuron, that's going to open the ion channels, and that's going to result in graded potentials. So graded potentials, they're just changes in the membrane potential. Those changes could eventually lead to an action potential, but not in every situation. The neurotransmitters effects are terminated by three ways. So either reuptake by astrocytes, which are a type of glial cells are support cells in the nervous system. So these are support cells that support the neurons, the nerve cells. Or reuptake at the axon terminal. It could also happen by degradation by enzymes, and then also by diffusion away from the synaptic cleft. All right, so this is going to be the end for this chapter, and we'll continue on the next one.